Hi, good morning, and welcome to the ZP Developer Zone. So we like to do this Developer Zone at least once a week, but with the volume of questions, we tend to these days have to do it twice a week. So with that said, I will go forward and just answer any and just essentially go forward on it. Okay, so today's 11th of February 2024, and we're answering questions that have come in during the week. It's probably worth saying that the entire technology stack at Zimmer and Peacock is really about getting data from sensors into the cloud. Whether you're doing this in a medical application or an environmental application, a wearable application, that's really what we're about. So it's either sort of from field to cloud, which would be kind of environmental, or maybe person to cloud, for example, which would be sort of medical and wellness. But everything we do is, we measure something on a sensor, and the data ends up, as I say, in the cloud. This is one of the hardware sets that I'll talk about today. This is probably most pertinent to question number 3. Question number 3 wants to make an IVD for looking at human health. They have a slightly unusual sample in that it's not sort of the normal blood or saliva or plasma. It's not those kind of samples. So I'll cover that as question number 3 today. But the kind of hardware that they're looking for is actually already covered at ZP, because we have what we call an OEM solution. By an OEM solution, I just mean, we may be the manufacturers of the original equipment, but then the person sort of rebrands it and builds upon that technology. It's probably worth saying that whether you're interested in measuring lactate or pH or glucose or making an IVD, we do have a webinar on the 27th of February. If a link pops up during this webinar today, I'll make sure I post the links underneath the video as well. This webinar, I know in that we do a live demonstration. We talk about our technologies for helping people to commercialize ideas. So please come along to that. It's probably worth saying that there's lots of ways of kind of getting material from ZP. We do corporate workshops where we come on site and actually do workshops with people. Tends to work with larger corporations where they can kind of get 20 people in a room, and that makes it kind of worthwhile. If you're a smaller institution and you want to sort of understand this technology more generally, I'm just thinking about some people who are asking about lactate at the moment. This would be a good workshop for them, in fact. So there's a workshop on the 4th and 5th of March. If you're interested in doing lactate monitoring and continuous lactate monitoring, this is probably the workshop for you. I'm also thinking about the people who are asking about pH and glucose. This is probably the workshop for you as well. And also the guy who's doing IVD, in vitro diagnostics, this is also a good workshop as well, because we do cover IVDs, and we do cover the commercialization of IVDs in that workshop as well. And honestly, I'll put the links underneath as well. I'll skip this today. We're just doing a conference in the UK in June, but no need to bear upon it. I mean, as I say, our whole philosophy is, at ZP we have we have a stack of technologies, because what we realize is, we recognize that there's people who need to achieve something, whether it's measure glucose, pH, lactate, and then there's people who want to deliver them that kind of technology. They could be clinicians, they could be entrepreneurs, and I always feel kind of frustrated, and not frustrated is too strong a word for it, but I realize that the way the funding is set up in this world, that in fact everyone is encouraged to reinvent the wheel. So if you're trying to make an in vitro diagnostic, you're encouraged to go out and build the hardware, even though these kind of platforms already exist. And you're probably encouraged to build the sensor from scratch, even though this is a pack of sensors with a QR code on it, even though these kind of technologies exist. And there's a sort of an encouragement to redo all of this, which is not really the philosophy, I would say, in the tech industry, where actually they're always stacking upon each other's technologies. And it's really because they actually understand that first of all, 90% of companies don't succeed, and I think they basically don't succeed just in this first instance because they're all trying to build this technology. It's probably more important. I saw there was a quote that I saw from Jeff Bezos just this morning. It's the execution and the adoption that's important. So people are so busy getting ready or doing the execution that they never actually test the adoption. So we're at ZP, we have a stack of technologies, and when I say a stack of technologies, I'll just put these up as well. These are wearable biosensors are obviously on a wristwatch format. I will discuss those in a minute, but we have a stack of technologies that means that kind of the execution is partially done, and then it's really seeing what the adoption is like, and that's getting that technology into people's hands. That's the really, let's say, hard bit. It's actually often the business case. So we're all about getting sensors into people's hands and hardware into people's hands as quickly as possible. It's all cloud connected. So if you gave let's say a lactate sensor for measuring lactate in saliva, for example, then you could give that fairly quickly and start getting people start to use it. Well, then what's nice then is it has a cloud connectivity to it. So that in fact, all the data is going to the cloud. You can check it, and it then allows you to actually, improve the technology. But it's best to be improving it whilst getting real world feedback. I don't necessarily mean paying customers, but I do mean real testers. There's such a delay in people having ideas and doing stuff and actually giving it to real world people. And with ZP, we've really solved that problem. It's a lean startup mentality, though. Lean is like, a sort of 10-year-old concept, so there's nothing super new there. But it still doesn't seem to be necessarily, certainly not understood by government, I would suggest. And it's not always understood by funders as well. Question number 1 today is about wearable lactate sensors. I'll discuss that. Question number 2 is about wearable pH and glucose sensors next to the skin, maybe in the sweat. IVD determination, or IVD technology, in vitro diagnostics that is, and also performing CGM experiments. In terms of lactate sensors, we do have a lot of formats. So this is the epidermis. Obviously, the epidermis is the top layer of skin. The lactate is residing sort of somewhere on or near the surface of the skin. So I've just made a quick model of the skin. We do have this kind of format, which I kind of call a wire sensor. I'll discuss this in a bit more detail in a bit. We do have this format, which is microneedles on the backs of these watches. In fact, I can just take it out. This is this insert is actually covered in microneedles, and those are designed to go through the skin. So I'll discuss that in a little bit as well. And we also have on sort of skin, non-invasive technology for monitoring the sweat, for example. So people are quite keen on doing that, doing lactate in sweat. I mean, we could do implanted, but I'm actually going to back off that because I realize this is a human application. We are doing implanted sensors, but we're mostly doing it in animal models. So I'll sort of back off that slightly. I would say that whenever you're doing a wearable, always think to yourself, is there a roadmap that I can have where actually I can first of all do IVD, in vitro diagnostics? I want to say it's an order of magnitude to do in vitro diagnostics as it is to do wearables. So it's kind of ironic, really. Here's an IVD, here's a wearable. This is much easier than this. It's, people raise hundreds of millions in order to do wearables, I would say, and take years. It's probably worth saying that we do have the ability to put lactate into this kind of form factor. This is something that, when I sort of say this, that a technology like this, we've easily spent 2 million euros in getting to this point, and this kind of technology is actually being implanted into animals. So if you're looking to make a kind of patch, this is not my product. This is the Abbott Freestyle Libre 2. It's a kind of famous CGM, continuous glucose monitor. They do have competitors. There's Dexcom and Medtronic, but this kind of technology is everything that's in there is summarized really in this kind of hardware here: the sensor, the power supply, the transducer for sending out data, the electronics itself. It's probably worth saying that the electronics that I'll talk about today, every time you see something with electronics in it, it's always the same electronic set from ZP. There's a real consistency across all of our different form factors. It's always the same hardware. So these kind of sensors, they're pretty robust with this sensor package, because actually this fish is not dead, it's anesthetized, and we're actually implanting these sensors into fish and monitoring biomarkers for understanding the stress that these fish are actually under. Still talking about lactate, but we're also able to put lactate into this kind of form factor. So this is an artist rendering. If you want to kind of get a sense of scale, these are the watches themselves. On the back side of the watch, there's a microneedle array. Microneedle arrays are quite nice because actually they allow you to do what we call, it's not that clever, it's allowed to do multi-analytes. I'll just unplug that again. So there's a little cartridge on the backside here that mates into here, as in the image on the right hand side here. This is a kind of quick video of just somebody putting it on their wrist. And yeah, so it's kind of, now it's probably worth saying at this point that this has actually had about 20 million euros spent on it. So that's why I kind of, it's not a small thing to make wearable sensors. I can talk about this because actually ZP owns this and we own the IP behind this. So this wearable microneedle technology has got something like 50 patents behind it, and ZP is the owner of those patents. And so ZP has what's good about us is we run a licensing model. So we are interested in working with people on these kind of analytes, but they do have to be well funded, and they do have to be able to essentially go all the way on this. So we are looking for people who generally can. And it's important because what it means is that this patent portfolio gives you what's called a freedom to operate. It can be extremely litigious doing wearable sensors, and so having a freedom to operate, i.e. something like 50 patents, or a license to 50 patents, is pretty important. It's probably worth saying, and I'm sure these people already know it, that we have been monitoring lactate on people in the sweat. This is an early set of electronics that we did. These days we've replaced these electronics with what we call the single-purpose biosensor circuit. So this will jump up quite a bit today, the single-purpose biosensor circuit, and I'll have links for it. But we were able to monitor them and send the data and sort of see the lactate. I should have labeled this up a bit better. But when this person started to exercise, then the lactate, or rather start to stress, then the lactate measurements came up quite a bit. I know I've said it already, but if you're going to get into lactate monitoring, you've kind of got this choice about doing wired sensors, which I would describe as transdermal, microneedles, which I would describe as minimally invasive, sweat, which I would describe as non-invasive. You've also got the choice to do IVD, in vitro diagnostics. It's a personal thing, but if you're not super well funded, or I would actually, if you want to get to market quickly, I would actually have a bit of a roadmap where I went IVD before I went wearables. But that said, we do have a very nice platform for doing IVD, in vitro diagnostics. This is also related to a specific question that's come in on IVD as well. But we do have this technology for scale. The picture that shows the meter there, this is that meter. The meter works with many of our biosensors, but also works with sensors that are maybe not developed today. But at ZP, we do have a really good range of screen printed electrodes. I was shocked the other day that somebody said, "Oh, you can buy your screen printed electrodes?" I was like, "Wow, okay." Yes, you absolutely can buy our screen printed electrodes. This meter talks to this app, and this app actually talks to a cloud system that we have called Julie. This is important because if you're sort of doing clinical trials or testing with people, it's really important that you get to see the data. And there's no need to be emailing that data or trying to find that data. The way the system works, the sensor goes into the meter, the sample goes onto the sensor. The sensor is told by the meter what to do. The meter gives the data back to the app, and the app uploads the data to the cloud, and then you get on their phone. And this is literally what the engineer was just doing a minute. Well, she's setting it up here, but you've got a concentration, but also you, and I'm talking to you as the developer or the entrepreneur or the academic, can actually see the raw data. You don't have to be putting patient IDs into this software, so you don't run foul of, patient identifiers like their name, their age, their sex. None of that is required. You just need to put a sample ID in there, and the sample ID is kind of allows you to be able to track the data later on. But what's cool is then you get the raw data up in the cloud, and actually you can just share it with your internal team, or you can also share it with a collaborator like ZP. So you can have these technologies distributed around the US, but there's not this fear of where is the data. They're basically alpha testers, beta testers, probably preclinical, maybe even clinical. And the question, where is the data? The data is actually just streaming in. So I'm supporting some people in India at the moment, and the data streams up to the cloud. And I'm supporting some people in California, for example, and the data streams up to the cloud. So it's really kind of a really efficient, modern technology base or technology stack. We also will call an API an application program interface. This is the glue that makes the modern world stick together. And whenever you make a payment or do anything online, often there's systems talking to systems, which is API, which is probably a good point. See, because I was saying earlier on that the problem that we have in the biosensor startup world is everyone keeps trying to build technology from scratch. You know, make a smartwatch, right? They build it all from scratch. Make an IVD, they build it from scratch. I get it. But in the world of tech, actually, no, they're all gluing, you know, services together using API, and we don't seem to be doing that in the environmental monitoring or the medical monitoring fields. But at ZP, we do recognize that people do actually integrate services and technology together, and that's why we got an API built into our system. This is relevant. Again, come to this webinar on 27th of February. Do one in the morning, one in the afternoon. So if you're in Europe, the morning or afternoon sessions will work for you. If you're in Asia, the morning session will work for you. If you're in the USA, the afternoon session will work for you. But it's definitely worth coming to this webinar to see this technology. And we have discussed this already. If these people want to essentially, I mean, working with ZP, you know, is paying us for consulting or paying us for development. If you're a pre-financed startup, then there's ways of starting on your own, which is you can get the single purpose biosensor circuit, you can get this wearable patch, and every time a link pops up, the link will be underneath the video. You can get yourself a lactate sensor, and you can also get yourself a test solution. And I realized there that says calibration solution, so I may have actually not linked to the lactate calibration solution, but we definitely have a lactate calibration solution. And probably by the time I put this on the YouTube, then I'll correct that for myself. Mental note. So my advice is, if you're funded, you can engage ZP directly. If you're not funded, then this list of hardware is probably the place to get you going if you're trying to build your own tech. Somebody's trying to measure pH and glucose near the skin. Again, I think what they possibly need at this point is they'll start with a single purpose biosensor circuit. My advice on the pH is you'll need the circuit, you'll need the patch, you'll need the pH sensor, which I can see I've got the right one here, and you'll need some test solutions so you make sure that you can essentially get some signal out of this. Similarly, if you want to do glucose next to the skin, you'll need the circuit, you'll need the patch, you will need the generation 1 glucose sensor. So it's the generation 1 glucose sensor, and I put a link here for you, so hopefully that will help you. And you will need the calibration solution. It's probably worth watching question number 4 actually, because I do talk about their testing glucose sensors, but that would be my advice for getting going. Otherwise, you can open a project with ZP and we can work with you, but if you otherwise want to conserve costs, you can do it just by buying these components. Question number 3 is about IVD. This is probably, I know I've said it already, but if people come to us saying wearables, I'm sort of, do you know how tough wearables actually is? I mean, just as a way, I've been making continuous glucose monitoring, CGM, for something like 15 years, and not in an academic sense. I mean, in an industrial context, I've been doing this. It's pretty tough, and I know you have to be well funded in order to do it. When people come to me and ask me for IVD, I think, well, right, that's a lot easier than trying to do continuous. So slight change of pace here. I'm going to run a couple of videos. One of the video in the top left-hand corner. So this person who was particularly interested in doing monitoring of material from human beings. The hardware, since we first talked to them, as advanced. The thing about ZP is we don't stand still. You know, it's an active business. You know, the engineers, we're always working on the next generation. So this is a platform we call Sense It All. It's pretty nice, in fact, because if you do get some material onto the platform and it needs to be cleaned, it's easy to actually dismantle it and give it a quick clean. In the other video, this person has a question about, well, the software development. The software development is pretty well done. So in a bit, if you're watching this video on YouTube, you'll be able to get your phone out, and if it's an Android, you'll be able to scan a QR code for Android, and if it's an iPhone, you'll be able to scan a QR code for iPhones, and then when you've done that, you'll understand that actually the software is already distributed. What's not distributed is because until we actually know exactly what your target is, and please don't tell us because there's not a disclosure agreement in place. Once we know what that is, then we can describe what's called a method file, and the method file is described. Then we can generate a QR code for it. So in fact, when I hold up this pack of sensors here, these are the cartridges, but here's a little QR code. So this will tell the meter how to run these particular sensors. So that's how we can tell a meter at any one time what to actually do. We have this kind of QR system. They were asking about costs on their disposable. I understand what they're asking. They're asking whether, their disposable at a volume of 10,000 units can be 10 cents. I can't answer the question unless I'm really engaged in doing a business model with you. So at ZP, we do business plans, we do business models, but I just don't want to guess on something, let's say. I will say that, the disposables that I'm quite used to, for example, today people can go onto our website, they can buy these screen printed electrodes, they can buy a pack of 300 off us, and they're about 89 cents each. Now that's a small pack size, and it's already 89 cents. So I can understand how I could get to your price points, but I haven't done the modeling, and we would need to be engaged in order to do that modeling. Just out of interest, if you're interested in those screen printed electrodes, here's the links. As I said, I was shocked the other day when somebody said, "Oh, you can buy screen printed electrodes from ZP." I was like, really shocked, but it obviously got a problem. We need to tell people about this. Now, if this person is trying to do an IVD for monitoring patients and potentially patients remotely, the things that we have in place are, we have the underlying screen printed electrode, but the screen printed electrode needs to become a sensor. So that needs to be paid for, because what I mean by that is an electrode is not necessarily specific. It can pick up signals, but there's no specificity about those signals. So there needs to be some work on the sensor itself. The hardware, if we can design and keep to this, is done. And what I mean by that is ZP is really good at electrochemical. So as long as the assay is, for example, electrochemical, then the hardware is done. The app is distributed on this store already. But what's not distributed is for each assay or each sensor, there's a particular QR code. And I'll bring this up in a minute. The QR code is put on the package. That assay is not developed because we don't know the target. So there is a gap there. But the database does exist. There is a database that any of our Sensei platform will send technology to the database. So the sensor will go into the hardware. The sample will go onto the sensor. The app, once the QR code has been generated for that particular assay, will be able to tell the meter what to do. The meter will give the data back to the app, and the app will send the data to the cloud. Now, the cloud system has been around for 10 years, so it's actually really developed in terms of it can receive data and turn data into concentrations, and the concentration can appear on the person's phone. So the clinician, the patient, they can get the number there and then. But you as the entrepreneur, the intrapreneur, the academic can actually get the data in the cloud. And that means you can really, keep an eye on the quality, make sure everything's working properly, especially in the development phase. And you don't need patient ID. It's really important to say that in order to receive that data. And you can then also share it with EP. So, we sort of imagine that people will come to us, we'll do some sensor and method development with them, we'll translate all that onto the platform, they'll start putting it in with real users, the real users will start using it, and then we can start getting the raw data. And that's this lean mentality that you've got to start finding out all your problems earlier on. And people spend too long rebuilding wearable sensors, rebuilding IVDs. They put that as the barrier. Well, it's not the barrier these days. And then they find out that they got problems. I'd rather find out problems much earlier on. In a sort of 3 to 6 month period is when I would like to start finding out I got problems. We do have an API built into it as well. And the API is because the technology world already, if you're running a cloud service, you'll already be running on an Amazon server or a Microsoft server or a Google server or an Oracle server. So people are already willing to run on each other's technologies, and they're doing it through APIs. It's just in the hardware world, the medtech world, that people don't have this mentality at this moment, but I think it will change. They did have some specific questions about their sort of sampling device, which was, they got a particular sample. It'll end up going onto one of our sensors. That sensor, by the way, slots into here. And it's not a slot, I should say. It just mates really well into the front of this unit. I'll show it in this photo here. So it goes there. So you're asking about cost on that. Until we've written a business plan or we're engaged in a program really hard. The underlying electrodes are 89 cents, but I can't really comment any more than that. The app is partially developed. So what I mean by that is if you have a smart device now, if it's an Android, you can scan this QR code. It will show you that the app is already on the Play Store. If you scan this, you'll find that it's already on the App Store for the iOS or the iPhone systems. Now, what's not developed is the method file to run the assay that you're otherwise going to engage us to develop. But you will see that once you've installed the initial app, the app will ask you, do you want to run a this sensor or this sensor? It's just asking for a QR code. And so for every time we develop a sensor or an assay, we're able to develop a QR code. So this is a really good way of suddenly distributing technology. And I'll show you that in a minute because I was going to walk through the technology, let's say, box in a minute. You were asking about cost. So it's probably worth saying that a single unit like this today, say you wanted 10 of these, then they're 1,699 euros, which, to be frank, in the medical world and many applications, it's really trivial cost. Now that's from us to you because that's extremely low volume. I'm going to put a link in a minute, so you need to really click on that link. This link will appear underneath this video. And it's worth saying that one meter is terrible because it sounds like a length, but 1 meter is 1,699 euros. I think a euro and a dollar today is pretty one-to-one. And I know I'm suddenly jumping to 10,000 meters, but 10,000 meters are 60 euros or $60 each. So I want you to, I need to go slightly up. Yeah, so go to this. You'll find that low volume, and people will be low volume for a couple of years. Low volume, it's, if you try to build a business plan. But these costs are nothing compared to the total development. The biggest cost I would say, in doing IVD development is not the cost of sensors and the cost of meters. It's actually the cost of people. Until you really have established a company and established the production, etc., it's really the people that's the costly part. But that said, we do have some costs on meters. And if you're writing a business plan, then you can use the drop-down menu here and sort of show that at 10,000 meters, you can get it at 60 euros each. The reason we're able to do this kind of stuff is because I have quite an extensive background in glucose sensors. So, we're quite used to making scalable technologies in terms of manufacturing scalability, but also in cost scalability as well. Now, the SenseIt is essentially our OEM platform. So I was just making a kind of unboxing here with, this is, in fact, I've got the box down here now. So just for scale, this is the box. But just saying, that at ZP, if somebody's trying to make an IVD, the SenseIt probably has it as an OEM solution, an original equipment manufacturer. We have a template for the manual, in that, it shows you what you get in the box. So we have the box itself. When you open up the box, there's actually a manual in there with quite nice, I would say, instructions on, what the person has. So this is a real kind of consumer experience. What's in the box, a little picture like that. It's kind of the kind of thing you expect. I know it sounds like a trivial detail, but honestly, these kind of details are the difference between easy engagement of technology and difficult engagement technology. So this technology, we call it Generation 4. And now, because everything's kind of been, we've been 10 years doing this for 10 years now. And, we've got good at this as a corporation, let's say. And suddenly, people are getting these boxes, they're just opening the manual and they're getting going. But it's taken us quite a while to get to that point. We always, this is a slightly different thing. We often offer training on something called food sense that we do. But suddenly found out that by the time we give training these days, most people have actually opened the box, gone through the manual and done it. this is an important part, that when they open up the manual, it also shows them where to get the software from. So I was talking about QR codes earlier on. Those QR codes that are in the, are shipped with the box. It tells them how to turn it on. There's a little button on the front there, and it's really quite super easy. As I say, most people everyone actually has already got going by the time we actually talk to them. It's nicely laid out in the box. In the video at the moment, it's actually showing the cartridges. So in fact, I've got the cartridges here. It's a bit bright, so you can't really see them, but we have the cartridges here. And the cartridges snap off. You put the cartridge in the sensor and you're kind of good to go. But on that, there's also a QR code, which I'm just highlighting my finger there now. That QR code, see, if we come up with a new assay for you, there'll be a strip of, let's say, sensors, and then there'll be a QR code that's unique to what you want to do. And that'll make the app essentially do what it needs to do. That's the meter itself, but you've seen the meter, and you can see it for scale here, so it's quite neat. In some assays, people require, so let's say it was a sort of, I'll give you an example. So a ZP, we actually help people measure, sounds ridiculous, but chilies and ginger. These are actually solid samples, and therefore we actually need to sometimes do an extraction, and we have, so therefore we're quite good at manufacturing materials and sending that material out, obviously in bottles, so there's the bottle for scale there, that's 500 milliliters, but we're quite used to making those materials, manufacturing those materials, and shipping those materials with the box as well. So that's, and I will just say that there will be a link in a minute, and that link will take you to, and there's the cartridges. They're quite neat actually, because all the sensors are in a kind of row, and you essentially snap them out. So it's kind of, there's some nice let's say features here, because it's sort of low form factor, so it's actually quite easy to ship. It's quite clear that it's quite intuitive that actually, these are in a row, and you can just snap them out. We don't have any problems with people actually getting their heads around that particular fact. As I said, there's a URL here, but I'll put that URL underneath the YouTube video at the end of this, so you can sort of go to that as well. So bringing an IVD to market, we think that the first phase, just making a minimally viable prototype, we think is about 500K. This is somebody who's super serious about doing this. I mean, you can bootstrap for a while, but realistically, you're going to spend this kind of money to get there. If people are really adopting our platform and they're able to stay on our platform, and that's, our platform is an OEM platform. We're not trying to be the brand owner, the customer owner on this. But if they're able to stay on our platform, then this is quite achievable. To really make an FDA cleared product and get all your paperwork and clinical trials in place, you have to be raising 5 to 10 million yourself. Now with ZP, this is a fraction of the cost that people normally have to raise. But we're just sort of assuming that you're going to run a sort of semi-virtual business. There'll be you, your stakeholders, a project manager on your side, but actually ZP will be doing a lot of the engineering, and then in that case you can do it quite low cost. It's probably worth saying that when people ask me financial questions, I get commissioned on ZP, rather gets commissioned all the time to write business cases, business models. So it is hard for us to actually not necessarily know all the details, but to then be giving out advice. So I can give out that kind of advice, but it's just advice, and we can't be held to it because we don't know enough details. It's probably worth saying that everything I've talked about, if you want to see this technology stack, and I think this technology stack will end up being your let's say go-to-market strategy, come to this webinar. Get your investors to come to this webinar on the 27th of February. I know it says March there, don't worry about that. The URL's been updated, and the link, and it will link you to a webinar. Come to the afternoon session. Last session, this came through YouTube. So at ZP, we talk about technology. We try to support the let's say the community a little bit. So somebody's asking about a glucose experiment. I think they're trying to describe a experiment where you're looking at a glucose sensor and you're showing that you've got signal or versus concentration. Signal versus concentration, I mean, you'll hear people talk about sensitivity, but you can basically calculate sensitivity. Now, I did get a statement from them, And they want to know, how do I extract the concentration I'm adding or injecting in a chronoamperometry experiment? So chronoamperometry, if you're not sure what that is, come to one of our workshops. They say they're injecting, or they're adding 0.1 molar concentration of glucose. I suspect it's glucose after each 100 seconds using a pipette when it's 25 microliters. I think, and it says, how do I label the concentration at each injection step? I think it's possibly how do they calculate the glucose at each concentration step. So first of all, let me say, I've done some calculations for you, so hopefully you can follow along. First of all, let me assume that you've got a sensor and you've got that sensor in 100 mils of buffer. 100 mils of buffer just means 0 millimolar glucose concentration. And let me assume that your stock solution, so this is the 100 mils of buffer. Let me assume that your stock solution is not 0.1 molar, it's 1 molar. I think this is a bit low. I know we may have said something about this on videos in the past, but these are just back in the envelope calculations I was doing for you. In fact, I put 500 here. That's interesting. Okay, well, maybe hopefully I'm right. I haven't made a mistake. I don't want to do 25 micromolar. I want to do 500 micromolar. Because I think if you add, I hope I got my calculations right, 500 micromolar of 1 molar solution to 100 mils of, let's say, buffer, you will create 5 millimolar steps. So let me just kind of run this a bit for you now. So I don't believe you've got a ZP glucose sensor, but if you had a ZP glucose sensor, we've turned it into a glucose sensor. You could potentially put that into a beaker. I'm just sort of suggesting 100 milliliters, and there's a little stirrer down here. And the first thing that happens is we test it in 0 millimolar glucose. So we get this kind of this break-in time here where the sensor's settling. And then, for example, you would add 500 microliters of 1 molar glucose concentrate, and that would give you 5 millimoles of glucose. Then you would have another 500 microliters of the 1 molar glucose concentration, and that would bring you to 10 millimoles. Then you would add 500 microliters of the 1 molar glucose solution to the 100 milliliters of buffer, and that would bring you to 15, and then you would repeat, and that would bring you to 20. So I'm sort of read your statement and think it's not quite the way I would necessarily do the experiment, and I then described that actually you need to increase the concentration of the stock solution, not 0.1 molar, but I've said a 1 molar, and I've said you're not adding 25 microliters, you're adding 500 microliters to 100 mil buffer. Some of you who are really good chemists out there will know that I haven't accounted for the dilution, but we're only adding 5, 10, 15, 20, 1, 2, 3, 4. We're only adding 2 milliliters into a total volume of 100 milliliters. So I know, within error, this will work out perfectly fine. I could do a more sophisticated spreadsheet, but it's just doing it very quickly. So in summary, I have read your statement. I think I understand what you're trying to do. I've told you to increase your concentration, the volume you're adding, and also told you what volume to have your experiment running in, and hopefully that will help you. As I say, I don't think this person's actually got a ZP glucose sensor. If they have, please send me an image. I'll be super interested. So question number 1, wearable lactate sensors. Wearable lactate sensors, there's the work with ZP route, or there's the start on your own. If you're pre-funding, then I would use the list of materials that I've sent you. This one from on glucose and pH, you can work with ZP, but that has to be funded, or you can use the list of materials that I've sent you, or I've put in the video, or you can do the IVD. This is question number 3, IVD determination in human material. You can, I think you'll be on this platform. I'd come to the webinar on the 20th, hopefully it's the 27th of February, and performing that CGM experiment. As I say, I've hopefully interpreted your question correctly, and then I've sort of recalculated what I think you should be doing. So I want to finish with a thank you. If you've got any questions of ZP, you can see that we do put a lot of effort into ZP. Best way of working with ZP is to open a budget with us and let us start working directly in a program. But otherwise, if you don't have a budget and you're pre-funding, then asking questions through the social media channels or through the contact us, and then we'll try and answer it at some point during one of these webinars. Okay, thanks very much.